بسم الله الرحمن الرحيم. السلام عليكم. خلينا okay, so we will be talking about the cytoskeleton and their involvement in this lecture. We'll talk about the involvement of one type of cytoskeletal protein, which is the actin microfilament in cell movement. So when we say cytoskeleton, we're talking about a dynamic network of protein filaments, okay? And these are found throughout the cytoplasm. There are three types. We have the actin microfilaments, as you can see here in red, okay? We have the microtubules, shown in the middle, and we have the intermediate filaments in here. Remember, we previously talked about intermediate filaments in the nuclear lamina, the nuclear matrix. Now, they have variety of functions, so they are responsible for the structural framework of cells. So the actin cytoskeleton, for example, is responsible for how cells look like. They determine cell shape and movement. They determine position of organelles, okay, whether they should be around the nucleus, like in the endoplasmic reticulum, or movement of vesicles in and out of the cells. They also determine the overall organization of the cytoplasm, as I said. Okay, so we'll start with the actin microfilaments. So these are also called just microfilaments. These are thin, flexible fibers, and they are thinner than the other two types of filaments. They are organized into different structures, and they can be complex structures as well. They can form bundles, or they can form a network, as shown in here. They form semi-solid gels as well, making the cell sort of like viscous, flexible, gelling, basically, so that it can change its shape and structure. They are regulated by a variety of actin-binding proteins as well. So we'll be talking about a number of these proteins, not all of them, because there are many of these proteins. Now, the actin microfilaments are abundant beneath the plasma membrane. They form a network of for cellular function, including, for example, vesicular fusion of vesicles, or fusion of vesicles with the plasma membrane. The actin microfilaments also stabilize the membrane structure as well. So we have 6 distinct actin genes. 4 of them are expressed in different types of muscles, and 2 are expressed in non-muscle cells. But overall, they have high homology, or that is similarity in. So the story starts with a protein known as the G-actin, and G stands for globular. So we're talking about just a single polypeptide that forms a dimer with another one, and then you have the addition of a third one forming a trimer. Now this process is known as the nucleation, and this is the initial step for the formation of the F-actin, where F stands for filamentous. Now, on both ends of this filament right here, you can have the addition of actin, but the filament itself is polarized, meaning that you have 2 distinct ends. You have an end known as the plus end, and the other one is known as the minus end. Now, the G-actin can be added on both ends, by the way, except that on the plus end you have addition of G-actin faster than on the other end. So what happens is that this G-actin protein is ATP bound. When it is ATP bound, you can have faster attachment of the G-actin to the filament. Okay. Now, the addition of an ATP, by the way, is not really required for nucleation, but the ATP bound ATP bound G protein is more stable. The filament is actually more stable, and it's faster in attaching to the filament. Now, what happens is that once it's added to the plus end, the ATP molecule is hydrolyzed to ADP, it becomes less stable, and there is a tendency for the G protein to, or the G actin, to be removed from the other end, from the minus end. Okay, so you have addition of, so you have G actin bound to ATP is added to the plus end, more probable than its release, and from the other end you have that is the minus end, where the G actin is now bound to ADP. There is a tendency for the protein to be released, okay, from the filament. Now this process, by the way, is known as treadmilling. Okay, treadmilling is actually treadmill هي هذه الآلة اللي الناس بركضوا عليها بالرياضة. Okay, this is treadmilling, or a treadmill. Okay, so what you see in here is that addition of G actin on one end and release from the other end. But so this process of actin filament formation, polymerization, stabilization, destabilization, depolymerization, and so on, all of these processes are controlled by a number of actin-binding proteins. We'll be talking about some of these proteins right here, including, for example, formin, tropomyosin. We'll talk about actinin, filament, fibrin. We'll also talk about cofilin, and so on. Eventually we'll talk briefly about alpha-ketinin, vinculin, spectrin, dystrophin, okay, and their functions. Okay, so you can always go back to this table and read it. Now, so as I said, the rate limiting step is nucleation, the formation of the trimer. This is the rate limiting step. Okay, this is the slowest process. Now, this process, by the way, is controlled by a protein known as formin. So from the name, you can tell what it does. It forms an actin microfilament. Now, you also have a number of proteins that are involved in the formation of filament and stabilization, destabilization, and so on, including tropomyosin. Tropomyosin is a protein that stabilizes the structure of the filament structure of actin. Okay, so that's what it does. Remember, we talked about tropomyosin before in the molecular biology lecture when we talked about alternative splicing. And we said that tropomyosin can be alternatively spliced depending on the cell type. So you have the formation of different types of tropomyosins. All of them are actin-binding proteins, but they have slightly different functions, kinetics, and regulation. So they're collectively known as isoforms. Tropomyosin isoforms. Now we also have another protein known as actinin. We'll talk about it also in a few minutes. Actinin is a cross-linking molecule. Okay, so it links two filaments to each other. Profilin is a protein that induces polymerization, extension of the filament, and opposite to profilin we have another protein known as cofilin, and what it does is that it induces depolymerization of the actin filament from the minus end specifically. So the actin filament filaments are organized in different ways depending on the actin binding proteins. Okay, so notice right here, for example, that we have different types of proteins: fibrin, actinin, which is a dimer, and we have filament. So, for example, you can have cross-linking proteins forming bundles of actin filaments, or you can have actin being organized more as a network. So you can see here in this electron microscopic image right here, where you can see actin organized as filaments, as shown in here, or also in the same image, you can have actin filaments organized as a network. So, talking about the actin bundles, in fact, there are different types of bundles depending on how close these filaments are. So for example, you can see that you have the protein fibrin, for example. It's a small protein and it forms a dimer cross-linking two filaments to each other. Now note the space between the filaments, it's very tight. Okay, so you find this structure mainly in intestinal microvilli. Okay, these fingers that extend out of the cell. Okay, and quite stable structures. On the other hand, you can have bundles as well, as you can see in here, organized in the same manner, except that the distance, the space between the two filaments is wider. And this is shown in here. This is formed by the protein alpha actinin, which forms a dimer. Now this structure here, you can find it more in muscle cells, allowing the protein myosin to interact with the actin microfilaments, inducing or regulating contraction of muscle. Okay. So note how these different proteins can form different. Now we have another protein, which is the filament protein. We talked about it briefly in 2 slides before. Now this filament structure allows for the formation of a network. Okay, so that's what it does. So the actin microfilaments can be organized as well, or they can be linked with the plasma membrane, okay, forming a cortex, so forming a network of filaments just underneath the plasma membrane. And the actin microfilaments can interact with the plasma membrane, and they can interact with the plasma membrane either directly or via proteins. Okay, and the actin cytoskeleton is best studied in red blood cells, and the reason for that is that the cytoplasm of red blood cells is much simpler than other cells. There is no nucleus, there are no organelles, and there's just cytoplasm. Okay. In addition, the shape of red blood cells is quite flexible, and they can change very easily. So they can squeeze in the capillaries, for example, passing through very tight areas. Okay, and they have to be smooth and flexible in order to move smoothly. So one of the proteins that form the cortex, the actin cortex, underneath the plasma membrane in red blood cells is known as spectrin. And spectrin is actually a long protein, and it interacts with the actin, okay, connecting actin filaments to each other. Now, spectrin itself allows actin to interact with the plasma membrane indirectly by interacting with 2 proteins. One of them is known as ankyrin, and the other is known as protein 4.1. So these 2 proteins can interact with plasma membrane proteins. Okay, so this is how you have this organization connecting the plasma membrane with the actin microfilaments. Okay, so it's indirect interaction. Now in muscle cells, you also have similar interaction between the actin microfilaments with the plasma membrane, or proteins in the plasma membrane, except that there is a different protein, and this protein is known as dystrophin, which is a very large protein that exists in these cells. Now dystrophin maintains the stability of cells, especially during muscle contraction, since it's a vigorous movement of muscle cells that can rupture cells, but dystrophin maintains the integrity of cells. Now, dystrophin, by the way, is linked or associated with 2 diseases. One of them is 2 muscular dystrophy diseases. One of them is known as Duchenne muscular dystrophy, and the other is known as Becker's muscular dystrophy. Now, these are caused by mutations, different types of mutations. Okay, Becker is caused by deletion, large deletion in the gene that encodes dystrophin. On the other hand, Duchenne muscular dystrophy results from frame shift mutation, from small deletion resulting in frame shift mutation. Now, Duchenne muscular dystrophy is more severe than Becker's muscular dystrophy. And someone might say, well, wait a second, but here we're talking about a large deletion. That's true, but with the Duchenne muscular dystrophy, what you have in here is a totally different protein as a result of frame shift mutation. On the other hand, right here you still have some regions of the dystrophin protein that can maintain its function. Okay, that's why the severity is more in Duchenne muscular dystrophy than in Becker's. And this is an X-linked inherited disease, by the way. So the gene is present on the X chromosome. Now, there's another way by which the actin microfilaments can interact with the plasma membrane, and this is known as focal adhesions. And you can see this beautiful immunofluorescent image right here. You can see in green the actin filament structures, and they end with these orange spicules or dots right here. That's the focal adhesion. Okay, so these here, the fibers in here, the microfilaments, are known as stress fibers, and they allow for cells to attach to the surface that they're sitting on. Okay, so what happens is that you have proteins known as integrins. Okay, and these integrins associate with the extracellular matrix, and at the same time they on the extracellular side and in the intracellular part of the protein, you have it interacting with actin-binding proteins, including proteins known as vinculin and taline. Okay, so you have these proteins right here in the focal adhesion. Okay, so anything that happens in the extracellular matrix, any movement, any change of interaction, can transmit a signal from outside to inside into the actin, so that cells can change their shape, or they can transmit a signal to other parts of the cell, including, if you remember when we talk about the nucleus, including the nuclear matrix lamina. Okay. Now we have other types of interactions that take place, and in this case we were talking about cell-cell adhesion or cell-cell contact. What happens here is that you can have a protein known as cadherin. So you have cadherin on both sides of two neighboring cells. You can have cadherins interacting with each other, and in the cytoplasmic part of the protein you can have cadherin interacting with another protein known as catenin, and we have different catenins, by the way, okay? Alpha and beta, but regardless, anyhow, and these interact with the actin cytoskeleton. So what happens in one cell can transmit a signal into the other cell via these cadherins and catenin proteins. So these are these structures are known as the Adherens junctions. Okay, so they form sort of like a belt around cells, these adherens junctions, with cadherins interacting with each other. So you have a sheet of cells عاملين زي الصفحة هيك interacting with each other. Okay, now you can have other specialized structures, by the way, that result from actin reorganization, and one of these structures is protrusions, يعني هيك تفرعات, okay, أو امتدادات. So you have these protrusions right here, and there are different types of protrusions. We have microvilli. So these are that's when you have actin organized as bundles. Okay, so we talked about fibrin, for example, as an example, and in microvilli. So these structures right here, the microvilli, you have bundles of actin filaments. You have a similar structure known as stereocilia, and you find this structure, by the way, in the auditory hair cells. These are the cells that are responsible for hearing. So you have these stereocilia can sense the sound, by the way, transmitting the sound from cells into neurons and etc. So you have these stereocilia also being organized in a similar fashion. Now you have another type of a bundle or a structure, a protrusion that looks like a finger-like, and it's known as filopodia. Okay. Also, you have bundles in cells, particularly when they move from one part to another. So right here you can have these are the filopodia right here. Now you have the actin being organized as what is known as a lamellipodia. So what you have in here, in fact, you have a network of actin cytoskeleton. So right here you have the lamellipodia, similar to that structure, by the way. So you can have a lamellipodia, and extending from the lamellipodia you can have the filopodia. Okay, you see this structure as well when cells are moving. So these are the microvilli, so we talked about them. So you have bundles of actin cytoskeleton. We talked about the also the coating that you see in here, the sugar coating of the cells, the structure known as glycocalyx. Okay, so this is the brush border of intestinal cells, the apical side of the cells. Okay, now so as I said, you can have actin being organized as bundles. We talked about fibrin as the actin binding protein that organizes these bundles, and note the tight spacing between the different filaments. Now we talked about protrusions, and we have different types of protrusions. We have pseudopodia, we have filopodia, and we have the lamellipodia. So the lamellipodia is when you have a network of actin organized looking like this. Filopodia is when you have bundles of the actin filaments as shown in here. And then you have what is known as a pseudopods. They look sort of like arms, and they are important for phagocytosis, for the formation of phagosomes when immune cells like macrophages try to eat bacteria and large particles. Now, lamellipodia, so let's talk about the involvement of the actin cytoskeleton in cell movement. So what you see here is a cell that is trying to move. It is attached to the substratum, that is to the surface that it's sitting on. Remember, right here you have the focal adhesions, right? The actin cytoskeleton attached to the cell surface to proteins like vinculin and talin, and they interact, and these proteins interact with integrins. Okay, remember that. So here we have these cells, and what you see in these cells is that as they move is that they extend the leading edge. Okay, they extend it, okay, and then what they do is that they start to change structure. They further attach to the substratum from the leading edge, and then all of a sudden they get released from the other end, from the tail. They get released and they retract. Okay, so it's like, so they move more something like a worm. So how is actin organized in that case? Well, what you see in here is that you have these focal adhesions that we just talked about, and when cells extend forward, they form a lamellipodia. Okay, the network right here. And then you have attachment, and then what you see in here is that contraction. You have detachment of cells from the other end, from the tail, and you have the actin microfilaments retracting or moving forward. Okay, so these are basically the steps of movement, and this is true for, by the way, slow moving cells like fibroblasts. But when we're talking about macrophages, for example, moving, they have loose attachment. They do not form these focal adhesions, by the way. Okay. Now, what are the proteins responsible for cell movement? Well, it's a number of proteins. Particularly, we'll focus on 3 of them, and these are small GTP binding proteins. Remember, we talked about some of these proteins in the previous lecture. They include RAS, RAB, RAN, the protein responsible for nuclear transport of proteins and other molecules, and RNA, for example. So we have what is known as the Rho family. And the Rho family particularly will focus on 3 of them. We have a protein known as Rho, and Rho is responsible for the formation of stress fibers. So what you see in here is this is just a cell grown without, just normal cell. This is when you have Rho overexpressed and activated in the cells. You can have you see the formation of the different stress fibers. When you have induction of RAC protein, you have the formation of the lamellipodia, okay, the extensions inside the cells. And when you have activation and overexpression of the protein CDC42, you have the formation of filopodia. So CDC42 is responsible for the formation of filopodia. So these are basically the steps. You have direction of movement. For example, cells are moving forward or in that direction. So we're looking at, we have a top view and a side view of cells. You can see cells form a lamellipodia, declaring or signaling that this is the leading edge, this is the head of the cell, this is the direction of movement, and you have the formation of a stress fibers on the other end. Now what happens then is that you have attachment at this end right here, the leading edge, and then you have retraction, detachment of cells from the other end, and these actin filaments get depolymerized, by the way, from the other end, and they extend forward.